Good morning, everyone. I'm really pleased to be here today, and as probably you understood from the title of the presentation, VDisk is a microfluidic platform for isolating extracellular vesicles from human plasma sample, and this is something I'm developing during my PhD at Hahn-Schickard Institute and Department of Microsystems Engineering at University of Freiburg. So in this talk, I'll present the characterization of the VDisk and discuss how it can address some of the key challenges in the field of liquid biopsy. So let's get started. When a volcano erupts, hot magma comes up from deep inside the earth, and as it cools down and turns into rock, it keeps some chemical clues which help geologists to understand what's happening deep inside the earth, and that's something they normally can easily see or reach. In a similar way, EVs or extracellular vesicles, which are nanoparticles 30 to 800 nanometer big, are continuously released by all type of body cells. They carry molecules like proteins and nucleic acids, which can reflect their parent cells. And these particles, at some point after going through the interstitial fluid, and so they end up somehow in the blood sample, and that makes the blood sample kind of a very rich source of EVs. And that means, theoretically, by getting access to the EVs in the blood sample, we can learn about what's happening inside the cells of our body, and that can be used for diagnosis of, for example, cancer, neurodegenerative disease, and some other conditions in a minimally invasive way. However, we might ask ourselves, why after almost 20 years of the research on the field, still we don't have any diagnostic tool based on blood EVs? And of course, the answer is not quite easy because there are so many complicated biological and technical hurdles on the way of translating EVs to diagnostics. However, regarding the plasma sample or blood sample, the complexity of the sample itself, which normally leads to co-isolation of the contaminants, as well as low abundance of EVs and heterogeneity of them, are among the main reasons. And beyond that, there is not really any standardized method for the isolation, and those existing ones normally suffer from low reproducibility and complex and labor-intensive process. So basically, these are the main challenges we would like to answer in our research. According to the literature, there are almost 10 to the power of 16 particles in just 1 ml of the plasma sample. And the most majority of these particles are like proteins, while EVs build up just a tiny fraction of this total particle count. And EVs coming from tissue cells and tumor cells are even less abundant, actually. And when it comes to the downstream EV analysis, the main contaminants which might interfere with the assay in the plasma sample would be low and very low density lipoprotein, high density lipoprotein, or soluble proteins. And ideally, all of these particles are supposed to be removed during the isolation protocol. On the other side, most of label-free EV isolation techniques rely on either size or charge of the particles for the separation. And as you can see here on this graph, all of these contaminants I already mentioned are overlapping with the EVs in at least one of these characteristics. That means if we really want to get access to some highly pure EVs, we need to combine these two features together. For example, if we separate particles initially by their charge, and then according to their size, it's quite likely that at the end we end up with a highly pure EV sample. And that's exactly how we developed our multimodal chromatography. So in the first step, we have a cation exchange chromatography step. We have these cation exchange resins, which we mix them with a plasma sample. And what happens in this step is that we remove the positively charged particles, mainly VLDLs and LDLs. And then we add the mixture of this resin and plasma sample into the second step. And here we have a sequential filtration with 2 membranes. The first membrane has a slightly bigger pore size to remove the bigger particles like platelets and also resins, of course. And the second membrane has a smaller pore size in the range of 20 nanometer, and that traps EVs while letting the smaller particles like HDLs and proteins go through. However, at some point, a kind of so-called filter cake takes place on the second membrane, and that reduces the efficiency of the filtration. And due to that, normally we co-isolate some part of small particles as well with the EVs. And that already reduced the purity of the sample. So to improve the purity further, we add the third step on our protocol, which is multimodal chromatography, or MMC in short. And for the MMC, we have this type of resins, as you can see here, which have an activated pore size. And normally, small particles like proteins can go through the pores, and due to their charge, they will be binded here, and they don't elute further. While EVs, due to their size, they don't go through the pores of these particles, and they can be collected after this step in a very purified way. So in the current version of the VDisk, we actually integrated or automated the step 2 and step 3. And for this automation, we took advantage of centrifugal microfluidics, which is very convenient to operate. And basically, the main advantage of such a system is that you have the centrifugal force generated by rotation as a main driving force for pushing the sample from a step to a step. And that already eliminates any need for external pumps or any other external pressure sources. So on the left side, you can see the picture from the VDisk after isolation of some plasma samples. And this cartridge has been made by injection molding, and then we seal the cartridge using a COC foil thermally. So the cartridge includes a sample inlet, 2 membranes, and both membranes are also integrated thermally to the disk, MMC column, and a big chamber for collecting the base liquid. And at the end, we can pipette out the purified EVs from this chamber. So as an example of the fluidics, for example, what's happening here is that the disk is just rotating at 25 hertz or 1,500 RPM. And then we generate enough pressure for pushing the sample through the membrane. And also here you can see now the liquid level is rising up and down. And the reason is we are switching between high and low frequency to generate a kind of mixing, so-called shake mode mixing. And that prevents the membrane fouling and improves the filtration efficiency. Also, what's happening here in this case, we are transferring the trapped and washed EVs from filter 2 into the MMC column by a vacuum generated using our waste liquid. So this mechanism has been presented by us last year at the MicroTAS, so I'm not going into the details, but if you are interested, please have a look at our preprint and our US patent on it. So, of course, when it comes to a new isolation method for EVs, always the first question would be that how high is the recovery of this platform or method? And that basically means that we need to measure how much of the EVs which are initially present in the sample would be recovered in the final isolate in an intact form. So to answer this question, we collected samples from a healthy donor and spiked the sample with the GFP labeled EVs. We processed the sample using the VDisk and also size exclusion chromatography as a kind of standard method for EV separation from plasma sample. And at the end, we measured the isolate using the fluorescent NTA on the same day of the isolation to prevent any freeze-thaw cycles which might damage the EVs. And according to the results, as you can see, VDisk achieved almost 84% of the recovery, and that was already much better than the SEC with a recovery of 61%. And also, we showed that we can just detect below 1% of the EVs in our waste liquid, which means not so many EVs are lost due to passage through the second membrane. To further investigate the reproducibility of this system, basically, we collected samples from 3 healthy donors and processed it again on the VDisk and SEC in parallel. And also to evaluate the robustness of our platform, we collected the samples from the same donors on the same day, but after a few hours that they consumed some fatty food, and we wanted to see how the increase in the lipid level of the in the plasma sample can impact the performance of the VDisk. So actually, in a lipemic sample, the appearance of the plasma looks like that. And this milky color is coming from the increased level of the lipid in the plasma sample. So then we analyzed the EV isolates from each method using various biomarkers, for example, CD81 and CD9 EV-specific markers, total protein, and total ApoA and ApoB100, which are lipoprotein biomarkers. So as you can see in this table, the CV values here showing the intra-donor variability of each measurement under fasting conditions. And VDisk actually showed a quite nice reproducibility, and all the CV values remain below 15%. And for SEC, we always had some higher variabilities, and even at some point, at some cases, as you can see, the CV values were up to 30%. And even processing the postprandial samples, we still saw that the VDisk can basically preserve or keep the CV values below 15%. And that indicates that VDisk can basically keep the robustness and reproducibility because it's working kind of in the same way in both fasting and postprandial samples. What you can see here is the efficiency of both methods in removal of the already mentioned contaminants, basically proteins, HDLs and VLDLs. And interestingly, both methods could basically remove the contaminants with the same efficiency in fasting and postprandial samples. However, as we saw in the previous slides, the variability of the SEC was slightly higher. Also, the particle size distribution, as you can see in these two figures, which is related to just one donor, is measured by nanoparticle tracking analysis, and that kind of remains the same for the VDisk in both fasting and postprandial samples. And at the end, to make sure that we can isolate EVs in an intact form, we did some cryo-EM microscopy, and we realized that the EVs are in the perfect shape, very comparable to the SEC, and they keep their double layer, and they're perfectly fine. However, until now, we always processed the VDisk using 1 ml of plasma sample, which was the initial volume we were aiming for when we started designing the disk. But at some point, we thought, okay, that would be really interesting to see how much changing the input volume would change the efficiency of this platform. And that's why we kind of repeated some part of the experiments using different plasma volumes. So 1 ml, 0.5 ml, and 0.1 ml of plasma sample. And at the end, all of these samples ended up in 700 microliter of the EV isolate. So the graph shows here that the yield of each method, independent of basically sample volume in all three variations here from VDisk, shows higher yield compared to the SEC. And the other two graphs here basically showing the purity of the EV as a signal to contamination ratio. For example, in this middle graph here, showing the ratio of the total EV to total protein, and in this case, it's a ratio of total EV to total HDL. And in both cases, we realized that the VDisk processing 0.5 ml of the sample is giving the best results. And what you can see here is basically the purity in terms of EV per VLDL and LDL. However, in this case, interestingly, 0.1 ml of plasma processed on the VDisk showed the best results. And all these measurements were also repeated with the CD9 marker, and we also kind of got the same behavior from the different variations of the VDisk. And also finally, to measure the concentration of the samples, we tested basically both methods using two different single EV analysis measurements, nano-FCM and pico assay with CD81 and CD9. So here I just show you the CD81 results. And of course, that's not a big surprise that we always get a higher concentration when processing higher, basically, volume of the sample. And always the lowest concentration is attributed to VDisk processing the lower amount of sample. And we always saw that sec concentration is somewhere in between this range. So as we saw in the last few slides, until now, VDisk offers a wide range of flexibility to meet different EV purification and purity and concentration requirements. And that already provides us with a big range of options for choosing which volume of the sample to be processed on the disk. So if we are aiming for some high concentration of the EVs, but not caring so much about the purity, of course, the VDisk processing 1 ml of the sample would be the best case. But if you are caring about the contaminants, of course, depending on which contaminant could be interfered with our downstream analysis, we can decide for 0.5 or 0.1 ml of plasma sample. So until now that we showed that this can, with a high efficiency, remove the contaminants from the sample and purify EVs, we went one step further and developed this next generation of the VDisk, which not only integrates the sequential filtration, washing, and multimodal chromatography, which we already had on the previous version of the VDisk, but also we added the plasma separation and cation exchange chromatography on the disk. And also, as you can see here, all the resins required for the whole process are pre-stored on the platform. And on top of that, we also integrated a detection unit, which allows us to be able to aliquot the EV samples directly after the isolation and kind of detect them using the detection unit integrated in our lab disk player in these detection chambers. For example, what we can do is to measure some EV markers directly on the disk, or even using this detector for doing some quality control of the EVs. So coming back again to the main challenges I discussed at the beginning, of course, we cannot really do anything about the complexity of the plasma sample. However, we showed that we can improve the purity to 2 times higher than what size exclusion chromatography can offer, and also with the improved recovery rate, with a very outstanding reproducibility and a fully automated process. And so altogether, we think VDisk could really shape the future of the EV-based liquid biopsy. And now we are quite looking forward to our next step, which would be using the VDisk in some clinical studies, which has been already planned, and we would be a part of it. So that brings me to the end of my presentation. I would like to thank my great team at Hahn-Schickard, also our project partners at University Clinics Erlangen and Freiburg, as well as our great and amazing prototyping people, which they always support us by manufacturing the disks. And at the end, if you are interested in our project, please follow our homepage. So thank you for your attention.